Good morning, everyone. Welcome to RAREfest. I'm Emma, and I'm one of the Cambridge Rare Disease Network team. Thank you for being here today. This weekend is about building a community of people living with rare disease, their families, researchers, scientists, all to learn, share experiences, and work together towards a better future for diagnosis, treatment, and support. We opened last night with a fantastic evening where our community shared their experience through performance, storytelling, and art. Today we have talks throughout the day, panel discussions, with hands-on exhibits in the room next door. So let's get started. Opening today, we have Rishi. Rishi's combined his lifelong love of science and a passion for music to found Singing Science. He composes musical mini lectures for students and general public. His background in genetics includes a decade as a bioinformatician and as a program manager for GA4GH, who develop and enable standards for discovery using genetic data. Today, Rishi is showcasing a specially adapted version of his show, Genomics, the musical. He combines basics of genetics and the importance of this for the rare disease community. Please join me in a warm welcome for Rishi, CEO, Creative Director, and lead performer for Singing Science.
Hi there, thank you very much for that lovely introduction. Yes, as has just been stated, so I am Rishi. I'm basically a failed rock star from the 90s who never got anywhere, tried to do my, you may have been one of the 10 people at my gig in 1997 at the Portland Arms, but probably not. So, but I have probably made a much more useful contribution towards the world in general through helping support scientists using by computer programming and so forth. And so this being Cambridge, I can sort of point out vaguely that way, I think. So 10 miles south of Cambridge is a place called Hingston, where you've got the European Bioinformatics Institute and the Wellcome Trust Sanger Institute, where I've spent time working. So, and the thing is, both those places just work, the work revolves around the genome and genes, and we just love genes so much that when we say hello in the morning, we said hello gene style, so let's have a go at doing that. So, the first thing to do to figuring out how to say hello gene style is to have a first look at a gene. So a gene there is like we can draw it as a series of colored blocks joined by lines. Sort of inside us, genes are really, really tiny things and sort of wrapped up really tightly on something called DNA. And that's a bit sort of messy to kind of draw as a picture, but we represent it out so we can understand it as a series of letters. And so bits of DNA will have like, thousands of genes on them. And we can think of genes as blocks of this DNA joined together to make a sort of a sentence with spaces between the words. But the first part of every gene is the letters ATG. So we can say hello gene style by saying ATG. So I'm going to say ATG everybody, and you say hello Rishi gene style, ATG Rishi. So ATG everybody. Thank you. All right, a bit of hellos there. So let's go for it gene style. It's great to have a human style hello, but let's do ATG everybody. Thank you. This is brilliant. So that's the so we're going to take you on a journey through genes, how we came to realize we had them inside us, what they do a bit physically for us, and then the role they've played in us being here today. So I'm going to start off with a question. What species are your relatives? So have any of you got like a brother, sister, child, father who's a banana? No. I was actually expecting more bananas because, I do this show in schools, and 100% of the children in the classroom insisted their parents were bananas, but that's another matter. Perhaps you've got a grandparent. You think, he's a bit quiet, but I've got to go and see him at Christmas, but he's nice to buy Christmas presents for because he just likes a bit of lettuce. Hang on, he's a slug. No? So we have this idea there that one species, when it has its next generation or its relations, they are similar species. So that's kind of one essence, something inside a species, when it makes its children, make sure it's the same species. But it's stronger than that. So dogs have been bred from the common wolf. That was a couple of thousand years ago. So What the wolf now, that's kind of a bit more vicious than what we have in our homes. It's more likely to bite your feet off than bring you your slippers. So the wolves that were friendly, though, towards humans said, okay, let's hang around together and become buddies. And, they bred them with other friendly wolves. And now we have something that's domesticated. And it's not just, behavioral, it's also shape and size. We have a whole range of shape and sizes and attitudes from dogs. But if you wanted to breed small dogs, you get 2 small dogs, put them together, and their child puppies will be, obviously small when they start out, but they'll grow up big to be small, if that makes sense. And it's not just with animals that we've been aware of this. We have seen this in the plant world. So if you go down the produce section of Sainsbury's or wherever, you'll find like a whole range of things that were all bred from the same starting point, a wild mustard plant. And so the kale that you buy, that was farmers saying, okay, let's have a look at this thing and see if we can get the leaves really, really big. So they found the kale plants with the biggest leaves, put it together with other kale plants, other mustard plants with big leaves, and eventually over thousands of generations, we've got these kale, big leafy kale that we can buy at the supermarket. And similarly with cauliflower and broccoli, the farmers bred the, let's have the one with the most flower buds, and that's why we've got so many florets on broccoli and cauliflower. They've been bred generationally, the generation-wise, the farmers picking out the ones with the most flower buds and breeding them together with other plants which have lots of flower buds to try and make something which has even more flower buds. And then this time next month, we're going to be celebrating Christmas Day. And then we can all thank the farmer who decided to pick the wild mustard seeds that would produce the most horrible, disgusting vegetable known to man. So we can think about that next month. But so people have had an idea for many thousands of years that there's some way of carrying the characteristics and traits down of one set of parents down to its children. But the person who really kind of started to examine this scientifically was this guy, Grigor Mendel. And what he did was he was a monk around in the late 1800s, and he basically bred 29,000 pea plants, and he carefully took the pollen off one after having measured its height, plant color, and flower color, and seed color, and things like that, and put it on the flower of another pea plant, again, which he'd measured, and he did this for many, many generations. Now, actually, he was working in a monastery. This was a thing lots of other people were looking at, but a lot of people were using sort of mice and rats to kind of do this together. But obviously, because he worked in a monastery, they didn't want the monks getting any ideas from watching them copulate, so that's how he got stuck with pea plants. So the first song I'm going to do is about his work. Grew up on the family farm, then moved to a monastery with an experimental garden. 6 years wrapping bags around the flowers of plants, giving us the music for the genetic dance. You must be Mendel. You must be Mendel, whoa, yeah. You measured traits of plants that grew from seeds, growing 29,000 and seeing how they got to breed. You then measured traits of plants that grew from their seeds, eventually proposing that living things had genes. You must be Mendel, 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 yeah, oh yeah, Mendel, Mendel, oh yeah. You must be Mendel, 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 yeah, oh yeah, Mendel, 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 oh yeah. It's a shame they didn't believe straight away in the theory you got from your plants. It was
was only after he had died that they gave the peas a chance. Growing 29,000 plants was not the only trick. You then had to perform some Mendel arithmetic, probabilities of dominant and recessive traits, giving us genetics and showing us the way. You must be Mendel, 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 yeah, oh yeah, Mendel, Mendel, oh yeah. You must be Mendel, 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 yeah, oh yeah, Mendel, 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 oh yeah, oh yeah.
Thank you. Cheers very much. So, the essence of what Mendel did was the starting point for science of genetics, and the real two takeaways he had was that we have discrete units that we now call genes inside us, and that is saying that if you have a tall plant and a short plant that get together, their child plant won't be a medium-sized one, it will still either be tall or short, so there's a fixed unit describing the height. And the second is that we have two copies of these genes inside us, because he realized there are situations where we had two purple-flowered plants, and that gave birth to a white-flowered child plant, happened in the situations where those two purple-flowered plants themselves were products of white and purple-flowered plants. So he, which makes sense a little when you look at the picture, maybe not with the words, but so he kind of put those two together, and that forms the starting point of the world of genetics. So let's have a look at what are the practicalities around the rest of this world of genetics. So first of all comes the question, where does the DNA, where's the genes live? And so that's inside our DNA, as we said at the beginning. So we have our cells, when there's sort of millions of them inside us, a bit like, you know, all the bricks in this building, and each one at its core has something called a nucleus. And the nucleus contains a repository of information, which it's like a library that we call the DNA. And that's this famous double helix structure. So this being Cambridge, I think I can point in that direction, 100 meters away. I should have checked this with Google Maps beforehand, actually, but I think it's that way. And you get to the Eagle pub where the announcement of the structure of DNA was made, though the plaque fails to recognize that the work was actually done by Rosalind Franklin to make this happen, rather than just the two blokes whose names are on the picture. So that's the DNA is this double helix structure, and we have inside us, it's stored in these pairs of what we call chromosomes, and so we have like 46 chunks of DNA in us, all paired up and tied together in a particular way. So let's have a look next at what DNA does and how genes are on it that allow us to say what genes do.
Body's made of organs, and our organs made of cells, and a cell has bits inside it that we call organelles. One of these houses the bit that makes us us. The genetic code is all wrapped up inside the nucleus. The genes inside us live on our DNA, coiled inside the nucleus. Protein templates. The genes inside us give a way to make RNA that sneaks outside the nucleus so proteins can be made. Genes The DNA is way too big to escape the nucleus, so it makes a copy called RNA that sneaks out with no fuss. It wanders round the cell, inspecting its new home, till something big grabs hold of it and says, "Hi, I'm a ribosome." The genes inside us live on our DNA, coiled inside the nucleus, protein templates. The genes inside us give a way to make RNA that sneaks outside the nucleus, so proteins can be made. Genes, DNA, RNA, genes, DNA, RNA, protein, genes, DNA, RNA, protein, genes, DNA, RNA, protein. Ribosome reads the RNA in bases of 3. Cat's a matching amino acid that happens to be floating past it, puts them all together into an amino acid chain. When the RNA is all used up, the protein has been made. The genes inside us live on our DNA, coiled inside the nucleus, protein templates. The genes inside us give a way to make RNA that sneaks outside the nucleus, so proteins can be made. Genes, DNA, RNA, protein. Genes, DNA, RNA, protein. Genes, DNA, RNA, protein. Genes, DNA, RNA, protein.
Thank you. Thank you very much. For I think Evan Reed is Doctor. Are you around here today? Oh, yep. He's going to be talking, I think, a bit about the actual process. But when he says, if he tells you that's not an accurate depiction of a ribosome, he's lying. That's a very realistic one, I'm sure. So yeah, that's the essence of what genes do. They kind of make proteins. Now, everything I tell you, the biology is always more complicated. There are actually many other types of genes, but we'll need a sort of 3-day music festival for that rather than an hour-long show. So what are some of the other things to kind of think about when we're considering the world of genes? So one is when we looked at Mendel and Mendel's experiments, they're very much, here's a physical thing that happened. We have purple plants or white plants, we have height. But we want to try and capture the fact that animals and plants respond in more complicated ways. So to use that, we call this phenotypes and phenotypic behavior. So it might be, if you're poked with a needle, you'll go, ow. Most of you go, but some people don't have nerve endings, so they won't go out. So you'd say that's a phenotypic response. If you see a big spider and you jump up, that's kind of a phenotypic response. Sorry, that one's for Kate, who might be watching in the video later on today. And then if I was to go out, tonight, out drinking, and then my wife is over there, then complains tomorrow morning, like, what's this mess in the downstairs toilet? I could say, that's my phenotypic response to drinking 10 pints of beer, So we want to capture things beyond just physical aspects of a being. It's how you respond to environment as well. And this is important because it can be then used to encapsulate how you respond to medicines and things later on. So there's a dog sneezing. Yes, allergies are one way in which you have a phenotypic response to pollen. Another thing that we need to think about is what we call gene expression. So not all your genes are always on all the time. So you've got 19,000 protein coding genes inside you. and you've got many different types of cells inside you. You've got kidneys and liver and muscle and your eyes. And all of those are actually very different cells, but they've all got the same DNA inside them. So of these 19,000 genes, not all of them are active at any one time. They can be, as I demonstrated there by a caterpillar and a butterfly, they're actually completely different looking organisms, but both of them have the same DNA and set of genes inside them. It's just some of them are turned on and doing stuff and some of them are not. And even plants, we can kind of show this using something called green fluorescent protein. So this is something we find in jellyfish, and it makes them light up a very bright green if you have a fluorescent light put on it. And what you can do is then try and figure out when genes are being turned on by putting this green fluorescent protein in next to a gene of interest and then seeing when it gets lit up. So you can investigate like other plants and animals using this mechanism. So here's a bit from the Green Planet that David Attenborough did last year, where he's simulating a caterpillar bite. And you can see there the cells are responding to a simulated bite by producing nicotine. So this is from a tobacco plant. And that green fluorescence protein has been put in to show that cells are activating their nicotine production to kind of deter the caterpillar, and also to tell other cells to start producing this stuff as well because they're being under attack. So let's have a little song now about green fluorescent protein.
Cell contains much DNA, but a decade ago it was hard to say, to tell which genes were being activated. Without obvious phenotypes, we would be frustrated. Let's face it, a cell is complicated. Trying to find out when and where a protein is made is hard, but Shimamura gave us our wish, copying the natural process in a jellyfish. There are times when we need to see if a cell encodes a particular protein. If we want to do that, we just tag the gene with the magic that is GFP. GFP. Green fluorescent protein. GFP. Green fluorescent protein. GFP. Green fluorescent protein. I want to know if there's expression of this gene. Get its output to glow so I can be seen. GFP. Yeah, GFP. Genes contain a stop and start. In between lies the active part. So to see if the gene is being activated for GFP, making sequence into these places, then let the cell get on with its life, doing its thing. Then by turning on a light, you can see just how much GFP shines. Where the protein has been made will be green and bright. GFP. Green fluorescent protein. GFP. Green fluorescent protein. GFP. Green fluorescent protein. I want to know if there's expression of this gene. Get its output to glow so it can be seen. Yeah, GFP. Yeah, GFP. Fluorescent protein. Green fluorescent protein. Green fluorescent protein. I wanna know if there's expression of this gene. Get its output to glow so it can be seen. Yeah, GFP. Yeah, GFP. GFP. Yeah, GFP. Whoa, yeah.
Cheers, thank you. So one of the reasons we've got now to genetics and being able to understand our DNA so much is thanks to these little things here. So we call these sequencing machines here, well, quite big things actually. There are these ones are usually made by Illumina, who are, I think they've got a thing there, so you can find out more about them there. But essentially what they do is allow us to sort of examine the genome of an individual, and also, just like we said, the green fluorescent protein allows us to examine if one gene is turned on and when it gets turned on. However, you often can't do that with people. No, no one's going to say, okay, yes, I'd love to light up in the dark so you can understand my genetic profile. No, but also genetics isn't simply one of the steps to understanding genetics. It's not just one gene does one thing. It's often a network of genes that get involved in a process and need to be turned on to make something happen. So you can actually get what's called an RNA profile by looking at the RNA. So depending on how you prepare a tissue sample or whatever you've got and feed it into the machine, you can then analyze different things about a being. So now we're kind of getting on to, we're all human beings, so we've all got something that's basically the same DNA. We started off with saying, genetics, genes encapsulate the idea that the next generation of a species is the same. But there's still something different. And that's because we have a group of genes that build the same set of things. So we'll all have overall the same sort of structure. But there will be letters that are different within a gene, and we call these variants. So there are different forms of genes, and they can result in different proteins. So if we think about the protein coding genes making proteins, it will result in a different proteins. And because those proteins go together to do things that affect us, will then either look different or can behave different or react differently to different things. So I'm going to do a little song for you now, and this one's called Variations on a Gene.
It's because of the genes inside us we grow up people and not like spiders. You may wish to dine on flies, but biology says that's not wise. Plants and animals, trees and bees, different species, different sets of genes. The building blocks are just the same. The instruction books build us in different ways. You and I are mostly the same. We all need hearts and we all need brains. Yet you and I are also different. That's partly due to variance. Variant genes have some bases changed, building us in slightly different ways. That's the way those genes behave, making us in different ways. I am me and you are you. It's the variants that help make this true. Same species, same gene set. Variant genes, though, lead to different effects. An altered letter in the DNA could lead to something not quite the same. It could be hair, it could be eyes, it could even make coriander something you despise. An altered letter in the DNA can lead to things not quite the same. I am me and you are you. It's the variants that help make this true.
Thank you. So I'm just going to have a brief look at some of that in a bit more sort of detail. We have the idea of different versions of genes inside us. You can alter a letter of the DNA. So there's something called OCA2 inside us that makes something called protein P. This is not the protein that makes you go to the toilet. Terrible naming, some of these biologists. And that makes melanin. And the amount of melanin then determines if you have like a brown-colored eye or a non-brown colored eye. Now, I say brown and non-brown colored eye because we have to start thinking about genes again, like I said, not as one gene does one thing, it's networks of genes. So if you don't have this overloading of melanin in your eyes color, it's a framework of other genes that will decide if you've got a green, gray, or blue eyes and things. So that is kind of just one of the effects there. To kind of understand sort of, this is how we look at genes on something called a genome browser. So they are, this is the ensemble genome browser that I worked on, but other inferior gene browsers also exist, should I say. You can see the blocks are actually quite tiny and the gaps between them are quite large. And sometimes you can actually get the gene being quite short as well, sort of compared to the full length of it. So we kind of touched upon what makes us different within a species, but we can't really move on without talking a bit about evolution. So evolution is, ultimately we have genes are instructions from building us into full-blown human beings or animals, and they affect how we can behave as well as how we physically are. And different instruction sets will build different end products. So like I like the idea of the Lego instruction books because they're just kind of like flimsy pieces of paper and the shape and things don't matter, but when you start assembling your Lego model, the physical structure of the protein starts becoming important and so forth, and you kind of get the different end product out of it. Now, it's important to realize, because you have the set of genes in you when you're born, you can't when you grow up, think, I want to be a horsey, and you won't magically change into a horsey. That can't happen. However many frogs you kiss, they're not going to sort of turn into an affluent future life partner. Though maybe the current economic climate may be a prudent thing to try anyway. There's a great place to kind of see lots of fossils that kind of led to, so let me think. So 100 meters that way, or maybe 200 meters if you go past John Lewis, you'll get to the Sedgwick Museum, which is a really great way, if you're sort of visiting outside of Cambridge, that's a really great place to kind of see the fossil record that they've collected up. But that kind of had people questioning around about the same time as Mendel was doing his work, asking why do we have things where species don't exist now, and also when the bones for those species were found, we didn't have human bones. So that sort of led to the realization that there was this process called evolution that meant new species had arisen. And the two people who really kind of sort of brought this to a full understanding, much better understanding of how the mechanism of it worked, and those were Charles Darwin and Alfred Russel Wallace. And both of them had been out to visit groups of islands, and that had let them have an overview of lots of similar species having adapted to the local circumstances of their island. So that tree there is what we call Darwin's finches, and he'd spotted these birds that presumably had a common ancestor, giving rise to different types of birds on different islands. So some of the islands were really rocky, and some of the islands had lots of trees. So you get some sort of woodpecker type finches on the ones with trees, and you got some ones with bigger feet that were better for walking on the rocks on the rocky type islands. So that kind of happened at the same time as Mendel did his work, and it's lovely that and the structure of DNA and understanding of our chemistry inside us have all joined together and sort of been united. So let's have a look perhaps at the process that he realized we went through. So he realized that what other people have not realized at the time was that, or thought at the time, was often an individual group of parents was trying, when it had its children, in some way to make it an ideal version for that environment. What Darwin and Russell Wallace realized basically is that it's more of a roll of the dice type thing. There would be mutants, and some of those would be better suited for living in that environment than others. So, and tying this to the genetics, we now know the mutation is kind of at the genetic level. You then have the new genes giving new effects because they will affect what type of proteins get turned on, made, or when genes get turned on and so forth. And then you have the pairing of it. It's really got to emphasize it's a two-step process, the mutation and then the natural selection, where the offspring with the new gene set survives and has its own children. So before we have a look at how some of those might affect us, let's see who likes Taylor Swift. Let's have a look at singers you like. Taylor Swift, yep, good fan there. What about Queen? I'm a big Queen fan. Yeah, okay, definitely. More older people there. And there was a 60s band called The Monkees. Anyone a big fan of them? Yep, so this is not quite the Monkees you're thinking of, maybe, but one of the differences between us and chimpanzees is that they can communicate and using less sophisticated mechanisms than we can. So, my wife's over there, I can say, I love you to her, whereas if it was a chimpanzee, I might do a poo and throw it at her, which probably, in our case means a relationship's probably irrevocably broken down kind of thing, but some people might go for that, I'm not judging. So why the chimpanzee is so important is because it's our most recent relative, our most recent common ancestor with another sort of major species is the chimpanzee. So 6 million years ago, there were a pair of parents and they had children, and one of those children gave rise to all of us in this room and all the human beings in the world, and the other one gave rise to all the chimpanzees. So this is... So we think we have a 6 million year time span in which we have evolved from something that was a more ape-like, or what we think of ape-like, so we are actually, technically apes, I think, into us. So what's one of the changes there? So I asked about singers because we have something, one of the changes there is something in what we call FOXP2. So that's a gene inside our brains that controls, helps develop the language center of our brains. And let's get that up. One of the, to examine, at a genetic level, how some of the changes that have happened are. You can see there's a diagram there showing three grey bars across different species of mammals, what the FOXP2 protein looks like. And in us, those two red changes at the top, those are two changes that have happened since the common ancestor with the chimpanzee that have allowed our brain to kind of develop a larger language area. Now, this is pretty amazing when you also go back even further in time and say, oh, actually, we're going to look back at not just the chimpanzee and us and this common ancestor. How has the FOXP2 gene been in other species? So you kind of look at the yellow block there is all the birds, and the FOXP2 gene in birds affects bird song and how they communicate. So this kind of really shows how genes can be built up into trees, not just that allow us to examine, for hundreds of thousands, by looking at creatures in the present and knowing when they evolved and so forth. We can put together this really lovely sort of tree of images across all the genes together to make this sort of gene tree type image. Now, this also does have the thing, we have genes in common with loads of other species. So how alike are you to a chimpanzee? We said 99%. So do you think a slug is more like a chimpanzee than, or less like a chimpanzee than a... Sorry, more like us than a chimpanzee, sorry. So we have 70% of a slug's genes are in common with us. So that's quite high, but we've got to do some things that are basically the same. We've got to process energy and things like that. So a mouse, will a mouse be more like us than a slug or less like us than a slug? More, yep. It's going to have a backbone and things like that. So we talked about if your relatives were bananas, would we be more like bananas? Would bananas be less like us than a mice? Yes, they're going to be less like us. And the tricky one is honeybees and bananas. So a banana's, say, got 50 to 60% genes in our, shared with us. What might a honeybee be? Do you think it's going to have less than 50 to 60% or more? More. So it is a tricky one. It turns out this is around 45%, so less is the correct answer. Well done there. Now, we're here today at RAREfest because we're talking about sort of a range of genetic syndromes. Now, I'm not an expert in this field. There'll be people giving talks and on the desks out there who really, really know their stuff there. But essentially, you can kind of break them down into sort of two types of things. Some that are inherited. For instance, sickle cell anemia is a case where you have two forms of a gene that results in a condition. For instance, one of the things this week was announced that Chris Hemsworth, who plays the mighty Thor, has done a show on Limitless, which I wanted to watch because he talks about how he's been identified as having 2 copies of the APOE4 gene, and this means he's got a higher likelihood of Alzheimer's when he grows older. It's also got the Hemsworth brothers all running around topless as well, so my wife wants to watch that for that reason. But she's blushing there. But yeah, there's a so sometimes things are inherited, and yet sometimes things happen in what we call de novo style. So they'll happen when an egg is either just pre or post fertilization. Something can happen that leads to different mutations in the DNA that are not, so you don't quite match your parents. And this is perfectly normal. It's powers, evolution happening. It's had, it will have happened to me and everyone in this room. It's whether the changes happen in particular genes, and there's sort of likelihoods of particular breakage points that things can happen. And the other sort of group of those is where you get additional chromosomes or so forth being brought into the process. So we're also interested in trying to solve these problems. So there's a great important thing here is databases. I love databases. I sequel with excitement when I hear about databases. Yeah, we've got some programmers, good. So you can have databases that allow, with rare diseases, one of the things is that's traditionally hindered a diagnosis is picking out, does it match anything else out there? And through the use of something like Matchmaker Exchange, you can have a system where you say this is my genotype and this is my phenotype and the symptoms. Is there anyone out there? And it could be, for instance, there's a case that we did at GFEGH where there was a child in Canada and they found 4 matches, 2 in Europe and 1 in Australia by requesting information across this database network. And what it allows is a broader, because we're talking about rare diseases, we're looking to try and find things from a bigger bucket of information. And we can link these databases up in a privacy-preserving way. That's a really important thing. You're not giving away patient information at the other end. You're just saying, I've got a match with these circumstances, and this was what the treatment path we followed. And in addition, we're now getting more. Genomics England, for instance, have a genotype database that they're building up, so to again allow for quicker diagnosis. And that would hopefully tie in with a more standardized genetic sequencing thing on the NHS. So one of the other things that I really love that proposes itself as a solution that ties in with the evolution stuff is the fact that we can look at other creatures. So cancers obviously is a genomic illness as well. It's related to when your genes go bad, but tends to happen when you're older. But there's a creature called a naked mole rat which doesn't get cancer. So I'm going to finish with a song about the naked mole rat.
I live beneath the grasslands of East Africa. In the dark, I'm pretty scary. I'm wrinkled and hairless with teeth that look like Dracula's. Yeah, I'm pretty ugly. We're the only mammals to live in colonies, led by a queen. Most of us end up being bachelors. She only mates with 3. genes, what can be gleaned for human beings. Woo-hoo. Naked mole rat, naked mole rat, naked mole rat, yeah. As I said, we don't have much sex, we've also got deformed sperm. But biologists' main interest is the fact we don't get cancer. You can give us 20 packets of unfiltered cigarettes, it won't be observed. So somewhere in our genes is a key to fewer deaths, so suddenly we're popular. You come into my habitat, burrowing into my genes. What can be gleaned for human beings? I'm a naked mole rat, and you've come into my habitat, burrowing into my genes. What can be gleaned for human beings? Naked mole rat, naked mole rat, naked mole rat, yeah. The hunt is on to establish just how I stay cancer-free. Biologists think it's because we've got a duplicate overcrowding gene. Most other mammals just have B27, which is similar in functionality to our unique B16, which gets turned on more quickly. I'm a naked mole rat, and you've come into my habitat, burrowing into my genes. What can be gleaned for human beings? I'm a naked mole rat, and you've come into my habitat, burrowing into my genes. What can be gleaned for human beings? Woo-hoo. Naked mole rat, naked mole rat, naked mole rat, yeah, yeah, yeah. Naked mole rat, naked mole rat, naked mole rat, yeah.
Thank you very much. Cheers. So thank you for coming along today, and I hope you get what you want out of RAREfest this year. I've actually realized I'm still using the same water bottle I got from RAREfest 3 years ago, so I've certainly got something out of previous visits. So there's only one thing left to do here, and that's to say goodbye gene style. So at the start of the gene, we always have ATG. At the end of the gene, we pretty much always have a tag. So I'm going to say tag everybody, and you can say tag Rishi. So tag everybody. Thank you very much. Cheers.